Oh, that's not what you need to see.
No, what, that makes a lot of sense.
Like, I need something.
No.
That's the. And I was actually, stuff the other day. Just sitting there, falls versus.
Oh, hey. I know, I'm sorry. I think so.
Okay, if you have not yet logged on and please make sure to, you guys have done a really good job of putting your real names. Often I do this and get real weird names and I'm like, I don't know who that is, but you're gonna get credit for your interaction today. So please, yes, definitely log on. On Tuesday, we studied pathways. And we had an in-class activity related to that hopefully everyone got a chance to complete. If you have not done that yet, please make sure you do so by next Sunday for that in-class activity. This is extra credit. So today we're going to do the Kahoot. If you complete the Kahoot, you get 2 extra credit points. If you win the Kahoot, you get 3 extra credit points. So let's make it competitive. Let's put some points on the table there. So today's our review session. I guess I should have led with that. We're going to be doing the Kahoot for today's review session. And then after we finish the Kahoot, this is about 30 questions long. You'll get kind of a sense of the multiple choice questions that you might be seeing on the exam. After that, I'm going to go over short answer questions. Questions you will see on the exam for your short questions, okay? I am recording this. I've recorded most, I would say 80% of the lectures thus far for this class, and I've started to create a YouTube channel for them, okay? After I leave today, I'm gonna post all of those lectures to the Canvas page. So if you're going through and looking at your study guides or filling out your study guides for maybe the first time, maybe who knows, you have an opportunity to go back and rewatch lectures if you so choose. Cool? Does anybody have any questions? There are some previous year's exams that are posted to our Canvas page, kind of at the very, very bottom. The way we see Canvas pages are different. I see a lot more stuff than you do. So it's near the bottom of the Canvas page. You can see some practice exam ones, but the keys haven't been posted. So have already put it on my to-do list to go back and dig and find those keys for those exams. So you have both the exam and the key. I'm going to go back, look at all of our previous quizzes, make sure they are keyed correctly, open them up for you to review. So you have previous exams, your own quizzes, your study guides to work through, recordings of lecture. Is there anything else outside of giving you the key for the upcoming exam that I can do to support you through this process? The Kahoot, we will go through it today, and then once the Kahoot is complete, If there's anything in here that you found particularly interesting or would like to continue to quiz yourself, I'll also post the Kahoot so you can go back and review it at your leisure as well. There's, like I said, it's only 30 questions. The questions in here are not too complicated. If you look through some of my previous exams, you'll notice that I kind of go through these weird phases of sometimes writing themed exams. I probably won't do that for this first exam for this class. But yes, so I'm gonna pause again and ask the question. Does anybody have any questions, concerns, requests from me regarding this exam? No, we're good? Cool, okay. Let's see, it's 2:05. I don't know, I feel like it's kind of quiet in here. We're gonna be playing a game. So why don't you turn to somebody sitting next to you and tell them about the best thing that happened to you this week? Okay, you don't even have to stand up. Just turn to somebody sitting next to you and be like, the best thing that happened to me this week was the sandwich I ate today for lunch. I don't care what it is, just turn and tell them about the best thing that happened in your week.
Not pretty good. After today. Actually, cool. Doing that.
Oh, okay, Cyrus is up there.
Yeah, they were like, yeah, one of those.
Very good friend, like. Yeah, my first year at UM.
Okay, you guys ready to do this? Okay, let's go. Let's go ahead and let's play some Kahoot. Okay. Oh. Film is the Kahoot. What? Oh. This is as loud as it goes. Which scientist is credited with founding the field of phrenology? Franz Gall. Woo! Sorry, Broca, Wernicke, they're linguists, right? Franz Gall is going to be our phrenologist here. Okay. In terms of knowing these individuals and exactly what they did, that's literally the level. I'm not going to be asking you dates. When are you ever going to use that information? But let's go like Gall, phrenology, Wernicke and Broca. Make sure you feel comfortable knowing what they did. And so, yeah, those are the big names. All of those other individual names, don't get too down in the weeds. Probably those three right there would be the ones that I would know. Just testing reflex time. Which molecule carries information away from the cell nucleus to the cytoplasm? The mRNA, yes. We've talked about the central dogma multiple times in this class. So knowing the steps of the central dogma and the molecules involved in those processes will absolutely be on the exam. And I'll kind of show you how in the long form question I'll ask you. This needs to go away. Okay. Good. Ben number 1 or Ben number 2? Which region of the neuron receives signals from other neurons? The dendrites. The axon is going to be the area of the neuron that is sending information. So yes. Which structure releases neurotransmitters into the synaptic cleft? Hmm. You could probably fight me on this one, honestly. You could probably make an argument for a synaptic vesicle being the answer to this question, for sure. It's not completely wrong. No, no, no. I think I think the question would have to be written more specifically, because as I look at that, I was like, what structure releases neurotransmitters into the synaptic cleft? Yeah, you could totally probably fight me on synaptic vesicle for this one. Okay. Stretch reflexes like the knee-jerk reflex are detected by which type of nerve fibers? Sensory. Are you okay? Do you have questions? Which scientist pioneered the study of language deficits after brain damage? Charles Sharon is a real. I think I might have made it up. Oh, I'm sure somewhere in history. I don't know if that dude did neuroscience though. Nice. Yes. Paul Broca. Golgi and Cajal are most well known for staining techniques and identifying cytoarchitecture. What are they? The central dogma describes the flow of genetic information from. Yeah. Good job, everyone. Big high five to the group. Maybe I should stop being so transparent about what I consider to be important. Kind of feels a little bit like cheating, right? You're like, don't stop that now. We're in this class. Which organelle contains digestive enzymes and breaks down waste material? It is going to be our lysosome. Yes. Yes, lysosomes tend to have a higher pH internally that help break down and degrade proteins. What does the endoplasmic reticulum do? I've lost control. Yeah, that's okay. That's what helps make protein. Thank you for answering my question. You're the only one listening. Everyone else. I'll just wait till the awkward silence settles in. Okay, you guys ready? You're like, yeah. Neurons transmit information through. Gotcha on that one. The great thing about Kahoot is it's more about testing reaction time usually more than anything, but then those questions happen. And you're like, oh, I got to read all the answers. So yes, A and B. Yeah.
Crazy.
Which scientist proposed the neuron doctrine? The audible F word was my favorite. That's been the favorite part of my week. Ooh, even spread there. It was Cajal. It was Cajal, yes. After he did the beautiful staining of individual neurons, he came up with the neuron doctrine that our brain is composed of individual cell units. Oh, making moves. Phineas Gage suffered damage to which lobe of the brain after an accident? Camping rod through the frontal lobe, That is true. Frontal lobe. Which type of glial cell provides myelin insulation for axons in the central nervous system? Central nervous system. It is in fact oligodendrocytes, which are a type of glial cell, but astrocytes and microglia are different types of glial cells, but oligodendrocytes are going to be our ones that are myelin producing cells. Does anybody have any questions on this? It's just kind of a pretty even spread here. You haven't given that information a glance. Whoa.
Some crazy.
Yeah, that was a big, who knew? How does a knee jerk reflex work? Blah, blah, blah. Ooh. 3 neuron pathway where you have your sensory neuron, your interneuron, and then your output of the motor neuron. I mean, you could make an argument for like a 4th neuron possibly, but in terms of these possible answers, it's probably the most correct of the 4. Action potentials are. Oh. Okay. I think we need to have a conversation, guys. Okay, let's have, so action potentials are always the same size. Graded potentials are graded and can be different sizes until they reach threshold. And once they reach threshold, then you have the action potential that occurs. Generally around 100 millivolts. It's generally like the range at which we're looking at for APs. APs are an all or nothing event that are always the same size. Cool? Yeah. Then number 2. Which brain region is important for speech production? Yeah, it's a tough one. If you get those 2 mixed up. Broca's. Anybody have any fun mnemonics to remember to keep the function of Broca and Wernicke's clear? Broca. Yes.
So Wernicke is responsible for like
Comprehension.
language comprehension. So the way that I think about it is like what. So like what means like I'm trying to like understand something, but I'm trying to understand it. I go, what is that?
Okay.
But that's how I do it.
Yeah. Okay, both good. Broca's is like more of an enunciation, a production. Wernicke's is more like, what am I
Like what?
hearing here? Yeah. Okay, so again, maybe that'll help you kind of keep it straight too if you have not thought about it that way yet. Endoplasmic reticulum is abundant in which part of the neuron? Endoplasmic reticulum. It hangs out in the soma. So most of our proteins are made in the soma, then they get trafficked to other areas. The only other place that you might be able to, like, again, make an argument is we do have some ER at the base of dendrites, but sometimes you also have free ribosomes there but a majority are going to be in the soma. Yeah. Man, but just kill them. Which type of scientific study best determines if a trait is heritable? Yeah. Family pedigree. I mean, again, you might be able to make an argument for sibling, but definitely family pedigree is going to tell you heritability across multiple generations.
Boom.
Which lobe contains the primary visual cortex? Or like, put this on the exam. Ooh. Occipital. Which scientific method assesses protein concentration? Concentration. Western blot. And number 2 back on the board. Okay. The synaptic cleft usually separates. Two adjacent neurons. Axon and a dendrite. We've talked primarily about axodendritic signaling. Probably the most correct answer here would be like a presynaptic and postsynaptic cell. That would probably be the best way to ask a question about the synaptic cleft and what it separates. Oof. What is the role of the PER gene in fruit flies? Circadian rhythm.
Yeah.
Woo! Go team. Who proposed the theory of localization of brain function? And it's not dyslexic. Yeesh. Woo! Even spread there. Okay, so Golgi invented a stain. Pahal used that stain. Broca was kind of the beginning of neuropsychology and was the first person to be like, this brain area does this specific function. So he's kind of the most modern, like, neuropsychological person that has like the structure function relationship kind of dialed in. I know it seems like we're going to need to know a lot about people and what they did. That's not true distribution of how this is going to happen. Yeah.
He actually drew out the different areas of the brain. Is that him?
That's Brodmann.
That's Brodmann.
Brodmann did the cell types.
They should have had different names.
They should have. I'll tell them that. Be like, those names are too similar. Yeah.
And
Yeah. Okay. What is the role of the clock gene in mice? We just saw this question. Yeah, circadian rhythm again. Oh my God. Brodmann mapping involved. Here we go. Very meta. We am examining cytoarchitecture. Okay. So Brodmann took brains, look at the different like cell types and mapped out the brain. Did not stimulate areas of people's brains. No.
Sophia Safaris,
The default mode network primarily involves the oh, wow. You're like, I kind of paid attention to that lecture. We talked about two things. One of them was the motor cortex, but the other was the prefrontal cortex and its ability to modulate the default mode network that helps us again kind of gives us the ability for introspection amongst other things. Okay. Where does the translation of mRNA occur? Translation, not transcription. Transcription does happen in the nucleus where you get mRNA and then that gets exported and gets translated via the ribosomes.
Why did they use
Because they knew biology students would eventually have to learn them. But the way that I remember, okay, so transcription translation, if this is tricky for you, think about it this way. DNA and RNA are both nucleic acids, right? So you're just kind of literally copying one nucleic acid to the other. And so transcribing is literally like writing. They're just keeping in the same language. And then it has to be shifted to protein, which is not a nucleic acid. It's a completely different thing. So it has to be translated into a different language. Does that help?
Yeah.
Okay.
I also, along those same lines, but I visualize like an old Christian monk, like actually like hand transcribing
Mhm.
the Bible, and it's like it has to be exactly that way every single time.
Mhm.
But then now it's in a bunch of other languages that aren't like Latin, so you have to make it not Latin.
Mhm.
And like that also works in my brain for the same reasons, but it's just helpful to see a little guy in my head.
Yeah, totally. Whatever makes the most sense, like use it. Absolutely. What ion is the most abundant in the extracellular cation? Ooh. Sodium. Just dominating. Okay. What limbic? Oh, the limbic system includes which structures? We might have hit this. In the temporal lobe, maybe that helps. Something. Hippocampus, amygdala, and hypothalamus. Yes. The corpus callosum connects the cerebral hemispheres. Last question. What's the role of microglia? Immune defense. Microglia are the immune cells of the central nervous system. Huh, I wonder who won.
Yeah.
Joe, nice job. Krista, great job. Wonderful. Ben, all the sleeper students from the back of the room coming on strong. Love it. Okay. Oh. Well, congratulations. Okay. Now we is that a frog?
Yeah.
It's a snake.
Oh.
Oh, I thought it was a frog. It looks like a snake to me.
I don't know.
I mean, way too. I like taking the scales off. Especially a Lily scale.
Definitely a lender. Oh, man, okay. I'm sorry, it's gonna get bright, no. Okay, so I will post that in Zoom for your review if you want to continue to use it to kind of test yourself. I would suggest looking at the exams though too. Before we switch over to kind of divulging the short answer questions, does anybody have any questions or concerns at this time? Okay. Short answer questions, not shockingly, based on how I've taught this class, is gonna be some drawing. So if you have a piece of paper, now might be a good time to pull out said piece of paper. And maybe as we're going through these, you can start to sketch through some possible answers, okay? Okay. Does anybody not have a piece of paper that would like one? I will keep one. Okay. Anybody want to be altruistic and give a piece of paper? Here we go. Okay, I got it. Got it? Okay, cool. Okay, so first question. English you. Okay. First question is this. Let's take ion. Yes. Areas, ions, let's see. Okay, so this question, that's a short version of it. What this question is gonna say is draw a neuron. Label the structures of the neuron. Things like axons, dendrites, somas, axon hillock, okay? And then also add in the ion distribution and core organelles and outline their functions. So this is a multi-parter. So what I've written up here is actually quite short. So in this, I do not care really about your artistic ability, and that's true for any drawing I ask you to do. I don't care, as long as you've labeled everything, I don't really care what it looks like. So we've got our neuron here. Okay. Here are our dendrites. Here is our soma. Here is our axon hillock. I don't know, man, in the past years, the axon hillock has been the thing that trips students up, and I don't know why, so I'm saying that out loud now, so maybe you pay attention. It's right here at the base of the soma where the soma connects to the axon. This is the axon. You get real crazy here. Say this is the presynaptic area. Area, okay. Any other area things? I mean, you can get real nuts here and draw in myelin. Draw in myelin. Anything else I'm forgetting here?
You can label them nodes.
The nodes?
Like those-
Pretty
Oh,
good. Yeah. I want to get fancy in French.
Is there-
Nodes of Granville. Okay. There and there. Okay. So next we have to label ions and relative concentrations and have, again, there is always going to be a concentration on the other side of the membrane, but where would we find a high concentration of sodium? Outside the cells. We're gonna go big sodium here, and if you want to, you can draw a little sodium on the inside. Okay, extension question, which way is that sodium gonna go? Into the cell. Okay, what other ions are we gonna want to capture here? Potassium, where's potassium high at? Inside the cell, which way does it want to go? Outside the cell. So again, the bigger lettering indicates a higher concentration. And that's also what that bracketing means, if you don't know that. Bracketing always means concentration. What other ions?
Calcium.
Calcium high where?
In
outside. Calcium actually has one of the highest concentration differential gradients. There's very, 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 very little calcium inside the cell because it's used as a signaling molecule for a variety of different mechanisms. And what's our last ion we should be concerned with? Fluoride outside the cell. I'm just gonna give you that one. Fluoride outside the cell. Okay. Yes.
I know that, am I incorrect in remembering that calcium is primarily localized kind of at the presynaptic area, because it has to do with, neurotransmitter,
I mean,
facilitates the release of neurotransmitter.
it does facilitate the release of neurotransmitter. We're gonna see it flux in there and specifically lead to the release of neurotransmitter. You might see like slightly small, we'll say heightened levels of microdomains of calcium influx there in higher calcium concentrations. But because it needs to be detected at such a small change in level, there are a lot of, we'll say, calcium quelling mechanisms in that space too. Does that make sense?
That does make sense.
Okay, yeah. Other questions? Okay, so we've done this. Last thing I have to do is organelles. Okay, we did kind of a general cell introduction, so let's start off with an easy one. What's this? Nucleus. What does it do? Holds DNA. Right? You can draw these guys. Okay, we have ER. We also, what also looks very similar to that, are Golgi. We can also have lysosomes. And I'm gonna draw just, what they look like don't really matter. I'm gonna draw these two little things. These are ribosomes. Okay, any other organelles? You could probably even just do this and be like the membrane. I would count that as an organelle. Anything else? Okay. Now, I'm just writing names of these organelles. I will be asking you for functions as well, so make sure you know what that organelle does. Okay?
Will it be more than 4 on the test?
More than 4 what?
Organelles.
No. There are probably like 10 organelles. I just let you select your 4 favorites. Okay? This is, you can, as you can see here, this is a beast of a question, but I'll tell you this. It is important for you. I feel like this is kind of what cellular neuroscience kind of is the backbone of. And you know what I do is every subsequent exam, I will ask you a version of this question, but we will continue to build upon it. So like the next section of this course, we'll be focusing on synaptic transmission, what happens between the pre and post synapse. So we'll ask you to draw a neuron again and go through this whole process and then continue to build in. Where are the ion channels at? Where is the presynaptic compartment? Show me transmitter diffusion. Outline the 5 steps of neurotransmission. So this is not something to sleep on now, okay? Because you're going to continue to see me harp on it again and again. Yes.
So we can draw it, we have to be like, it's not for you.
Whatever makes the most sense to you, honestly. Like I said, I don't really care about your artistic, prowess. What I'll probably do is just make this like part A, part B, and part C. And if by part C, this drawing is so nuts, you're just like, I can't draw it in here. You can just probably go ahead and over here go like, oh, number 2 is nucleus. And just kind of draw a line to where it's generally found. I don't really care about the representations of the organelles. Does that make sense?
Yeah.
Does that answer your question? Okay, cool. Any other questions? Okay. How do you want time? Plenty of time. Okay, does everyone have a piece of paper? Ish, okay. 2, I'm gonna have you guys draw the knee-jerk reflex. Okay. Now, I'm not gonna go through the whole knee-jerk reflex. If we can draw it together right now if you want, but you just did this in class, right? So hopefully you feel pretty confident about your knee-jerk reflex drawing. There's a couple representations pretty clear in the slide decks. Again, I do not care about your artistic prowess. Just literally draw the line in. You have your. I'm like, I'm not gonna do this. Now I'm doing it. This is me drawing a knee. This is me drawing a cross section of our spinal cord. Okay? Hammer, sensory information going in. Synapsing here, splitting out. This is interneuron. Here are your motor neurons. They're going to be innervating that musculature. So that's again, my level of artistry, but I will ask that you kind of probably label that a little bit more intensely. Ooh. You guys ready for your next question? Okay, next, let's go. Next question. I've been harping about the central dogma. Outline the steps of the central dogma and the processes, okay? So what I mean when I'm saying that is the thing that we just discussed, which is DNA make, I shouldn't give you the answers. What comes next? Makes. Okay. I won't take off points for spelling because I can't tell you how many times I've misspelled protein and how many Brians I have in notes too. Anyway. Nope, not even a chuckle, brain. Nobody else does that? Okay. What's this process? Transcription. I will ask you for this. And this is translation. Okay. The extension of this, again, is kind of choose your own adventure. This is the central dogma. We spent an entire lecture talking about methodology. Identify one method and where it hits and what it does in this within the central dogma. So you're going to do this plus one method. Okay. So examples would be like, oh my God, you go PCR, but I'm going to ask you to explain what exactly happens in PCR, very generally. Protein, you could do Western blot. And again, very briefly describe what you're measuring there. In that case, it would be things like protein concentration. Or you could go ICC, immunocytochemistry, or IHC, immunohistochemistry, and that would be localization of protein, okay? I don't need you to outline the protocol for me for these methods. I just need you to tell me what part of the central dogma it's assessing and what it's telling you. Just thought of another long form question, but I want to ask you it. Remember about temporal and spatial localization? That will be a multiple choice question. I've already in my very long format sketched out my long answer questions here. Okay. You guys ready for your next question? Okay. Explain a social behavior via genetics. Okay. So in our, we did again an entire lecture. I like to try to make these very open-ended and choose your own adventure, whichever part of this lecture resonated with you, right? So like the PERN clock gene, that's an example of a behavior. I said social behavior, but an example of a behavior that is determined by genetics. So you could explain to me how that gene kind of infers that behavior. The example of the voles, the prairie and montane voles, another example of a behavior that is determined via genetics. The rover and the sitter genes in, was that Drosophila? Yep. Larvae, that's another example of a behavior that's explained via genetics. So I'll just ask you to kind of explain a little bit about those experiments. Okay. And again, this is kind of choose your own adventure. You get to just choose whichever of those experiments resonates the most with you and explain them to me. Okay. Ooh. Last long form question. These are pretty involved. So you're only getting 5. Outline a pathway of your own choosing. Any pathway. Remember yesterday we did the exercise? I had you come up with your pathway, explain a pathway. You could do the sensory pathway. You could do the motor pathway. You could do the DMN. You could do the visual system. Just choose a pathway, right? That's what you did on Tuesday, is you chose a pathway and put together a slide. Am I wrong? That happened? Or is going to happen? So that is the fifth question. So just to run down those 5 questions again with multiple parts to them. Number 1, draw a neuron, plus the ionic concentrations plus 4 organelles and those organelles functions. Number 2, draw the knee-jerk reflex. Number 3, draw the central dogma and explain a method for one of the steps of the central dogma. Number 4, explain a behavior that we can, outline a experiment where behavior is dependent upon genetics. And number 5, outline a pathway of your choosing. Okay? Those are, I'm not, I kind of want to call people who take my class. Ben, I'm not joking. Those are going to be the questions on the exam, aren't they? Yeah, I'm not. He took 260 from me. I do the same thing in that class. I know there are faculty who are just like, read the textbook, know every word, good luck. But no, I'm not that person. I'm telling you exactly what you need to study in 6 feet on this exam. I will give you those previous exams and their keys. You can see how I write my questions, the types of things that I focus on. Look at the study guides. I often look at the study guides as I'm writing the exam too, because I consider that to be almost be like a contract between like, I'm making you note this in a study guide. It is important enough for me to do so. And so that's what I will assess you on. Okay. I don't, again, not everybody believes in curricular transparency. I do. So hopefully you guys will hopefully benefit from it. I know going into the first exam with a new faculty member can be tough. That's why I'm giving you old exams from previous years. But in the meantime, between now and next Tuesday, if you have questions, if you have concerns, if it gets to be Monday and your anxiety is eating you alive, reach out to me. I'm happy to meet you for an office hour. If I get multiple questions, I'm also happy to do a review session. That's like not a problem at all. In addition to that, the other resource that you have is there is a discussion board that is posted on this week on our Canvas page. So if you're studying and you're like, did she say this was important? And you want to post that into the discussion board, you can also go there and give feedback to your other members of the course. Okay. So I feel like I've given you all the tools, hopefully to succeed. Does anybody have any questions or concerns before I let you go for today?
Yeah. Do we need a blue book or a Scantron?
Do you need a blue book or Scantron? Wonderful question. If you want to know, I'm gonna give you my honest answer here. The answer to that is no, because I work for the pharmacy school and they buy Scantrons for their students, so I'm just gonna jack 30 of them and bring them for you. So you guys pick enough question. Okay. Brilliant. Scantron with this we have love. Yes, so I will provide Scantrons for you. It does expedite the grading process. I'll be grading all of your exams, your short answers by hand, so I see what you know. I will be having my PA do that. Oh, the last thing I'll remind you of, your study guides are due. Okay, so if you have not done any study guides yet, now is a great time to start filling those out. Okay, so just make sure you attend to that in addition to studying for the exam. Okay. Okay, good luck. Have a good weekend. Please get out, enjoy the weather, get sleep, drink water, take care of yourselves, and I'll see you all on Tuesday. Goodbye.
This week.